Welcome back. In this web lecture, we're going to continue on with the general topic of how animals respond to their environment. In the next two web lectures, we're going to focus on the role of the endocrine system and how it coordinates with the nervous system to produce both long-term and short-term responses to internal and external stimuli. So much of this will be sort of a review and application of concepts you learned way back in Chapter 11 on cell signaling, and we'll see how we can apply and expand on some of those ideas. So chemical signaling in animals involves the release of molecules into the system. And so this will be in response to external stimuli or internal conditions, and this release of molecules can be done either by the nervous system or the endocrine system. These chemical signals are going to produce longer-term responses than the rapid messages transmitted by way of the nervous system through action potentials. So action potentials are going to be very quick and very short-lived. These chemical signals are going to be slower to initiate and last longer. The particular kind of chemical signals that circulate through the body fluids and affect distant targets are known as hormones. The endocrine system is a group of organs and cells that produces chemical signals and secretes them particularly into the bloodstream. So it's going to be the system that's involved with secreting these hormones. The nervous system and the endocrine systems often overlap in function. Again, the nervous system can produce a very rapid response that's short-lived. The endocrine system can follow up by maintaining those changes and maintaining that response over time. So let's just review cell-to-cell signaling. This is material that we covered back in Chapter 11. Animal chemical signals are present in extremely low concentrations, but can have enormous effects on their target cells. Remember, these signals are distributed to the whole body, so in any particular cell is only going to receive a very small number of them, but we see mechanisms for amplifying their effect. So unlike action potentials, the messages that chemical signals carry have a very long-lasting effect. So in combination, electrical and chemical signals allow animals to coordinate the activities of cells throughout a relatively large body and to create relatively complicated behaviors, such as all of the different changes that are necessary for mating behaviors, for aggressive behaviors. All of these are going to be coordinated by both the nervous system and the endocrine system. So animals use these chemical signals to communicate throughout their body in diverse ways. Communication between the cells and animals through these secreted signals can be classified based on two primary criteria: the type of secreting cell, the route that's taken by the signal in reaching its target. How is it going to get there? So let's look through the examples of different kinds of signaling pathways. So first, autocrine signals. So these signals act on the same cell that secretes them. So the secreting cell is going to be identical to the target cell. Molecules are going to arrive by diffusion. Paracrine signals diffuse locally and act just on nearby cell. So like autocrine signaling, the message is going to be transmitted just through diffusion in the local environment. Endocrine signals are hormones carried between cells by blood or other body fluids. So here we have hormone-releasing cells that just secrete into the extracellular fluid. There are blood vessels nearby, so these cells are going to be surrounded by capillary beds so that they are going to diffuse right into the bloodstream, be carried throughout the body, but remember, they're only going to affect target cells that have the appropriate receptors for that particular hormone. Neural signals, the chemical signal that we're talking about here is through neurotransmitters, diffuse just a short distance between neurons. So the particular signaling cell in this case is a neuron, and the mechanism for reaching the target is simple diffusion. We can also have something called neuroendocrine signals, and these signal molecules are referred to as neurohormones. So they're hormones because they're distributed by the blood, neuro because they're secreted by a neuron, and so in this case the axon terminals communicate directly with a blood vessel and secrete those neurohormones into the bloodstream, where they're distributed, again, system-wide, but only responded to by cells that have the correct receptor proteins for that particular hormone. So now let's take a look at the different kinds of pathways by which hormones can act. So some endocrine cells respond directly to environmental stimuli by secreting their hormones. So this is very simple. They detect the stimulus directly. The same cells that detect the stimulus secrete the hormone into the blood, and the blood carries them to the target cells. Pathways with an additional step. In these pathways, information about environmental conditions is first gathered by sensory neurons, then integrated and processed by the central nervous system before the production of hormones. So in this case, we've got a stimulus that is received by some kind of sensory cell, sent to the central nervous system, where it's going to go through any number of different interneurons along their pathway to be able to properly integrate and process that information. And then some neuron in the central nervous system is going to release neurohormones into blood vessels nearby that are then going to be distributed throughout the body, affecting their target cells. A third, even more complicated path is that the released neurohormones stimulate other cells in the endocrine system to produce hormones. So here we have a sensory cell receiving a stimulus being integrated by probably a large number of neurons within the central nervous system. A neuron in the central nervous system releases those neurohormones into the bloodstream, where their target cells are going to be another endocrine organ that is then going to release its hormone into the bloodstream, finally circulating and finding those effector cells that are going to respond to it. Hormones fall into three major classes of macromolecules. They can be polypeptides, steroids, or amines. So the polypeptides and most amines are water-soluble, so they're going to be hydrophilic. Steroid hormones and other largely nonpolar hormones are going to be lipid-soluble. They're going to be hydrophobic. So as we saw when we looked at cell signaling, these are going to have very different properties in terms of how they interact with their targets. So here we have water-soluble polypeptides such as insulin, and a water-soluble amine such as epinephrine. And then we've got steroids that are generally hydrophobic such as cortisol, and then thyroxine is an example of an amine that is also lipid-soluble. So let's just review the different properties of these different kinds of signaling molecules. So water-soluble hormones are secreted by exocytosis. So they're going to be packaged in these little vesicles. Remember, they don't cross cell membranes very readily, so they need to have another pathway to getting out of the secreting cell. So again, these are going to be stored in little membrane-bound vesicles that will then come and fuse with the cell membrane and become part of the cell membrane, releasing those contents from the interior into the extracellular space, where they will diffuse into nearby blood vessels. Lipid-soluble hormones, on the other hand, can move very readily through cell membranes. So they're going to diffuse straight out of that secreting cell across the cell membrane and then diffuse into nearby blood vessels, where they're going to bind to certain proteins called transport proteins. This keeps them dissolved within the blood until they diffuse out of the bloodstream through the membrane. And then when they reach their targets, they are going to diffuse directly through the cell membrane of their target cells, and they're going to find their receptor proteins within the cell. And these receptors can be found either in the cytoplasm or all the way inside the nucleus. So let's first look at the response pathway for lipid-soluble hormones, because it's a little bit of a more simple, direct pathway. So the response to a lipid-soluble hormone is usually a change in gene expression. Remember, most of these hormone receptor complexes function as transcription factors. So when a steroid hormone binds to its cytosolic receptor, a hormone receptor complex forms, and that is going to move into the nucleus. There, the receptor part of the complex, so think of the hormone as just activating this receptor. It's the receptor part of the complex that's going to act as a transcriptional regulator of specific target genes. So it can be either an enhancer or an inhibitor of transcription, but it's going to change gene expression. So let's take a look at a specific example of this response pathway for a lipid-soluble hormone. So the steroid hormone receptors that bind to estrogens in particular are very well characterized. In female birds and frogs, estradiol, which is a form of the steroid hormone estrogen, binds to a cytoplasmic receptor in liver cells. So remember, this hydrophobic steroid hormone is going to diffuse straight through the cell membrane, where it's going to find its particular receptor protein and bind with that receptor protein to create a hormone receptor complex. So once this is bound, the hormone receptor complex is going to move into the nucleus, where it's going to activate the transcription of genes needed to produce egg yolk. So here we've got our transcription factor. Remember, the transcription factor is part of this receptor protein that's been activated by the binding of the hormone. This is going to attach to the DNA at some promoter region, and it's going to trigger the transcription of mRNA for a protein called vitellogenin, which is needed for producing egg yolk. And then again, those transcripts are going to move out of the nucleus and be translated by these ribosomes into the protein vitellogenin. So steroid hormones can cause changes in mRNA production by a large number of genes. So steroid hormone receptors have a distinctive DNA binding region called a zinc finger. Remember, this site where the interaction with DNA takes place is on the receptor protein, not on the steroid. So this is on the steroid hormone receptor. So that zinc finger portion of the receptor protein is what makes physical contact with the DNA at that promoter site. So here we see the hormone response element on the DNA that's interacting with those regions on the hormone receptor complex to begin transcription. The steroid hormone receptor complex binds to that hormone response element and causes changes in gene expression. Remember that as long as this complex is bound, transcription is going to continue. So each complex is going to lead to the production of many copies of this transcript. So in this little cartoon, they only have room to draw one RNA polymerase attached to this little bitty DNA, but in real life, there would be a whole bunch of RNA polymerases transcribing this at the same time, producing a whole bunch of mRNAs. And remember that every single mRNA, a whole bunch of ribosomes can attach to the same one to produce a whole bunch of individual proteins. So remember that a single steroid hormone attached to a single receptor can result in an amplification of that signal into many, many, many proteins being produced. So now let's take a look at the response pathway for water-soluble hormones, these hydrophilic molecules. So remember, it all starts with binding of a hormone to its receptor outside the cell membrane. So remember, this hormone never actually passes through into the cytoplasm. It's going to cause a change in that receptor protein. That's going to be what transfers that signal, that information from outside the cell to inside the cell. The actual response of the cell can vary. It can lead to responses in the cytoskeleton, so some kind of shifting around of materials within the cell or change in shape of the cell. It can be enzyme activation, so it can start some kind of metabolic process, or it can also be a change in gene expression through the activation of transcription factors. So for example, the hormone epinephrine, also known as adrenaline, regulates many organs in response to stressful situations. So epinephrine is going to bind to receptors on the plasma membrane, for example, of liver cells. So here we've got our epinephrine coming and binding to a G protein coupled receptor, which again, remember, is going to use a GTP to activate that G protein, and in this case, it's going to activate adenylyl cyclase. This triggers the release of messenger molecules that activate enzymes and result in, ultimately, in the release of glucose into the bloodstream. So it's going to go through this whole pathway, and then eventually the end result is going to be the breakdown of glycogen through the second messenger system. So what is the second messenger? So one effect of epinephrine is to increase the glucose levels in blood. Epinephrine activates an enzyme called phosphorylase. This is the enzyme that cleaves glucose molecules off of glycogen. Remember, glycogen is a whole big repeating string of glucose monomers. And it's going to do this by triggering a signal transduction cascade that has the activation of this phosphorylase as the ending step. So phosphorylase exists in an active and an inactive state, and it switches to the active state when it becomes phosphorylated. So when a phosphate group is added here, a monomer of glucose ends up being cleaved off of a glycogen molecule here on the other side of this enzyme. So here we've got our string of glucose monomers all join together into this glycogen polymer. When we activate the phosphorylase, it's going to release all that glucose, and we know that epinephrine is the signal that causes this to happen because we can do experiments in which there's no epinephrine versus epinephrine added, and we see that there's a much higher level of active phosphorylase. So we can be pretty sure that this is the pathway by which this happens. And the key molecule in the activation of this phosphorylase is our old friend cyclic AMP, which acts as a second messenger to amplify the signal. And it does this through a phosphorylation cascade. So when epinephrine binds to its receptor, it activates the G protein that then activates the enzyme adenylyl cyclase, just as we saw before back in chapter 11. Adenylyl cyclase then converts ATP to cyclic AMP. Cyclic AMP then initiates a chain of events called a signal transduction cascade by binding to cyclic AMP-dependent protein kinase A. This enzyme then phosphorylates the enzyme phosphorylase kinase, which phosphorylates phosphorylase. So this whole chain of phosphorylation, which we call a phosphorylation cascade. Thus, cyclic AMP transmits the signal from the cell surface to the signal transduction cascade, and together, cyclic AMP production and the subsequent phosphorylation events amplify the original signal from epinephrine. Remember, every step along this cascade, these are enzymes that are capable of catalyzing this reaction over and over and over again, and we actually saw this specific example of amplification when we looked at epinephrine back in chapter 11. So a single binding of a single molecule of epinephrine may trigger the release of millions or even billions of glucose molecules. Here's a graphic showing how this happens. So epinephrine binding to the receptor, activating the G protein, which is going to slide along the membrane until it finds adenylyl cyclase. There it's going to convert ATP to cyclic AMP. Cyclic AMP is going to activate cyclic AMP-dependent protein kinase A, again using ATP. The protein kinase A is going to activate the phosphorylase kinase, which is in turn going to activate phosphorylase, which is ultimately going to be responsible for releasing all of that glucose from the glycogen molecules. And remember that amplification happens during each one of these steps, so there's going to be an exponential increase. So now let's take a closer look at this phenomenon by which the same hormone can have different effects in different kinds of cells or even at different periods during development. So epinephrine has been studied in several mammals to understand the cell surface receptor and the transduction of the message. So results have shown that epinephrine binds to two different kinds of receptors, alpha and beta receptors. And it turns out that there are 4 distinct types of epinephrine receptor, and each is found in a distinct tissue type, and each induces a different kind of response from the cell. The same hormone interacting with different kinds of receptors is one of the ways that epinephrine can trigger a completely different response depending on which cell it interacts with. Another way that this can happen is through the signal transduction process. So signal transduction cascades are diverse and complex. And so even using the same basic pathway, for example, using cyclic AMP as a second messenger, cyclic AMP may activate a completely different kind of phosphorylation cascade that has a completely different effect in a different cell. So target cells that have the same receptor protein may have different second messengers and different enzyme systems, or any combination of these. So the same hormone and receptor can give rise to different responses in different target cells just because of the assemblage of proteins and pathways that are contained in that cell. So again, this helps to explain one of the most fundamental observations about hormones, that the same chemical messenger can trigger different responses in cells from different organs or in cells at different developmental stages. So cells contain different receptors, second messengers, amplification steps, protein kinases, enzymes, or transcriptionally active genes, and that's going to determine how they respond. This explains why hormones in particular are so effective at producing system-wide coordinated responses. If a whole suite of different responses from different systems are needed to respond to the exact same stimulus, the same environmental situation, this activates all of those systems with a single message from a single messenger. So this is a very efficient system for coordinating whole body responses. So let's take a look at the composition of the endocrine system. What's in it? So organs that secrete hormones into the bloodstream are called endocrine glands, and the tissues and organs that make up the endocrine system vary widely among animals. So for the most part, these endocrine glands are made of a special kind of epithelium called glandular epithelium, but there can be other kinds of tissues that also function as glands. So for example, neurons that manufacture and secrete hormones are particularly important in insects where they regulate molting, metamorphosis, and other processes. So nervous tissue can be an important tissue in this endocrine system. In addition, salmon have an unusual gland that secretes a hormone responsible for regulating calcium ion concentration. So that's an example of kind of a unique organ that's possessed by just one particular taxonomic group. In addition, there's incredible diversity among the endocrine glands just within the human endocrine system. So cells in many other organs and tissues secrete hormones even though they're not organized into discrete glands. So you can have individual cells in a completely different organ that's going to be secreting these hormones to the bloodstream. So for example, there are individual cells in the stomach that are going to secrete hormones even though they're not part of an actual gland. There's another kind of gland known as an exocrine gland. So exocrine glands differ from endocrine glands in that they deliver their secretions through ducts into a space other than the circulatory system. So whereas endocrine glands are surrounded by capillary beds so that they just secrete their hormones into the extracellular space and then they diffuse into these capillary beds. In the case of exocrine glands, there's an actual duct that delivers these secretions, for example, into the digestive system. So one good example of a single organ that functions as both an endocrine gland and an exocrine gland is the pancreas. So the pancreas secretes insulin, a hormone, just into the extracellular space around it, and that insulin is distributed through the bloodstream. But the pancreas also has a little duct that leads to the duodenum, that first segment of the small intestine, and it delivers digestive enzymes and other substances directly into the digestive tract. And so in that digestive function, the pancreas works as an exocrine gland. So here's a summary of the different glands and their functions within the human endocrine system. And I don't want to go into too much detail because I don't want you to actually memorize all of these glands and all of these hormones and all of these functions. But what I do want to point out is this is color-coded by the different kinds of hormones that are released. So the vast majority of these are polypeptides, short chains of amino acids. The sex hormones are nearly all steroids. So these are nearly all those hydrophobic messages that tend to work as transcription factors. And then a very small number of them here in purple are amino acid derivatives. So mostly protein type substances are being used as these hormones. So now let's take a look at the different kinds of hormone pathways and the ways in which they're controlled. So hormones are assembled into regulatory pathways, and these can be simple or they could be more complex. So in a simple endocrine pathway, hormones are released from an endocrine cell, travel through the bloodstream, and interact with specific receptors within a target cell to cause a physiological response. So this is the most simple kind of pathway. We can also have a simple neuroendocrine pathway in which the stimulus is received by a sensory neuron, which then stimulates a neurosecretory cell. And that neurosecretory cell is going to secrete a neurohormone, which then enters the bloodstream and travels to target cells. So this is the simplest case of a neuroendocrine pathway. So let's take a look at an example of a simple endocrine pathway. So for example, the release of acidic contents of the stomach into the duodenum. Remember, that's the first segment of the small intestine directly connected to the stomach. And in the stomach, the substance being digested has been mixed with a lot of hydrochloric acid to help the breakdown of those stomach contents. And so by the time it gets into the small intestine, it's very, very acidic. So the acid contained in that digesta moving into the small intestine stimulates the endocrine cells there to secrete secretin. So this is an example of individual endocrine cells within a different kind of organ, this time the small intestine. So here we have our stimulus, which is a high hydrogen ion concentration, causing this endocrine cell directly to release its hormone. So this hormone is going to circulate through the blood and arrive at target cells in the pancreas, which is a gland that's located right behind the stomach. And the pancreas is going to respond by secreting a substance called bicarbonate, which is highly basic. It's going to bind up those protons to neutralize the acid. As a result, the digesta in the small intestine is going to be less acidic. It's going to produce less of that stimulus, which is going to cause less of this hormone to be released. And as you know, we call this a negative feedback regulatory loop. So negative feedback loop inhibits a response by reducing the initial stimulus, thus preventing excessive pathway activity. So when the pathway is not needed anymore, it's shut down, and it's shut down directly by the response. Now let's take a look at a simple neuroendocrine pathway. So for example, the suckling of an infant, a nursing infant, stimulates signals in the nervous system that reach the hypothalamus. So the stimulus is going to be that direct tactile stimulation. It's going to be integrated through the hypothalamus. The response of the hypothalamus is going to be to release a hormone called oxytocin from the posterior pituitary gland. So then the oxytocin, again, is going to circulate throughout the body. Its target cells are going to be located in the mammary glands, and it's going to cause the mammary glands to go ahead and release that milk to the suckling infant. And in this case, the release of the milk is going to stimulate more suckling by the infant, which is going to actually increase that stimulus. And so this is an example of positive feedback, which is going to reinforce that stimulus to create an even greater response. And this will continue until the infant stops suckling, and that will shut down that stimulus directly. Now let's think about how the endocrine and nervous systems are coordinated. So in a wide range of animals, endocrine organs in the brain integrate the function of the endocrine system with that of the nervous system. So that's where these neurosecretory cells come into play that are secreting these neurohormones. So as we'll see in vertebrates such as humans, this has a lot to do with this interaction between the hypothalamus and the pituitary gland, which are located directly adjacent to each other, sort of in the bottom of the brain. Here we can see where that's located in the skull, and here is how closely they are situated to each other with the hypothalamus and the pituitary gland sort of dangling directly beneath it. So first let's think about this in invertebrates though. So invertebrates have an exoskeleton, this hardened outer shell that prevents them from being able to grow unless they actually shed that outer shell and secrete a new one. So this is what we call molting. It's also known as ecdysis. So here we see, this is kind of a weird thought if you're not used to it, that the sort of outside appearance of an insect is actually that exoskeleton that can just be shed off. So what you end up with is this ghostly representation of the animal, of just an outer shell. The animal itself has kind of escaped from it and secreted a new shell. So the endocrine pathway that controls this molting of insects as they grow, particularly in the larval stages before they've undergone sort of a transition like metamorphosis, as we see in things like butterflies, this is going to originate in the larval brain, where neurosecretory cells produce a hormone called PTTH. So in the prothoracic gland, PTTH directs the release of a hormone called ecdysteroid. This is a very awkward word, but remember that ecdysis is another term for this process of molting. It's a steroid hormone that promotes ecdysis or molting. So bursts of production of ecdysteroid trigger each successive molt and also influences the process of metamorphosis, changing into the final adult form. Metamorphosis is not triggered until the level of another hormone called juvenile hormone drops. So let's take a look at how that works. So here we have our insect brain with the neurosecretory cells sending signals through these different regions of the brain, but releasing this hormone PTTH, which is going to have its target in the prothoracic gland, which is then going to be the gland that actually releases this ecdysteroid that has the direct effect of causing molting. So in these early larval stages, when they go through their first molts before metamorphosis, this release of ecdysteroid is going to be accompanied by a high level of juvenile hormone coming from another region of the brain. The ecdysteroid is going to promote molting, but the high level of juvenile hormone is going to prevent metamorphosis. So as we go through additional molts and additional phases in the larval stage, we continue to have release of ecdysteroid, which is going to promote molting, high levels of juvenile hormone. But as we go to the later stages of larval development, we're still having the release of ecdysteroid, but the levels of juvenile hormone are going to start to decrease, and a low level of juvenile hormone is going to allow the molting into the pupal stage, but it's going to initiate metamorphosis. And so finally, metamorphosis occurs due to the low levels of juvenile hormone, again under the direction of ecdysteroid, to allow that final molt. So in vertebrates, it's going to be the hypothalamus, this portion of the brain that coordinates everything about endocrine signaling. And it's going to receive information from all the sensory inputs and all the integration centers, and it's going to initiate the appropriate neuroendocrine signals. And it's going to do this through a gland that's sometimes referred to as the master gland. This is going to be the one gland that controls all of the other glands. So this is located at the base of the hypothalamus, and it's called the pituitary gland. So it's going to be, remember, this little gland that's hanging down from the bottom of the brain, and it's composed of two fused glands, the posterior pituitary and the anterior pituitary. So these are part of the same gland, we call it the pituitary gland, but these two separate but fused glands actually have very different developmental origins. So the posterior pituitary actually develops as an outgrowth from the hypothalamus. So you can see that the neurons actually are continuous all the way down into the posterior pituitary. The anterior pituitary has its developmental origin as a glandular epithelium. It originates from the epithelium of the roof of the mouth that grows in toward the brain and ends up fusing with the pituitary. And so this has a non-neuronal origin, and it's going to function in a very different way, as we'll see. So the posterior pituitary stores and secretes hormones that are made in the hypothalamus. These hormones actually travel through the axons from the hypothalamus down into the posterior pituitary gland where they're stored. The anterior pituitary makes and releases hormones under the regulation of the hypothalamus. It's going to release hormones that are secreted by the posterior pituitary under the direction of signals coming from the hypothalamus. So neurosecretory cells of the hypothalamus synthesize the two posterior pituitary hormones, and remember these are going to travel through the axons and be stored down in the axon terminals waiting to be released. And these two neurohormones are antidiuretic hormone, ADH. We've seen this one before. It's very important in regulation of water levels, but it also has a role in regulating other aspects of physiology and behavior. The other hormone that is produced by the posterior pituitary is oxytocin. Oxytocin, as we have just seen, regulates milk secretion by the mammary glands, but it also has a lot of effects on behavior, particularly in pair bonding and bonding between a mother and infant. The anterior pituitary hormones are a little bit more diverse and complicated. They control diverse processes such as metabolism, osmoregulation, and reproduction. And hormones secreted by the hypothalamus control release of all of the anterior pituitary hormones. So for each anterior pituitary hormone, there's going to be a releasing hormone coming from the hypothalamus that's going to cause the release of that hormone. The hypothalamus is going to release a neurohormone into a capillary bed here in the hypothalamus. That blood is going to be delivered directly to another capillary bed in the anterior pituitary through these vessels called portal vessels. So portal vessels are defined as blood vessels that move between one capillary bed and another capillary bed. And so those releasing hormones are going to be distributed to the anterior pituitary through the blood, where they're going to stimulate the release of hormones So, for example, prolactin-releasing hormone from the hypothalamus stimulates the anterior pituitary to secrete prolactin, which we see here, which has a role in milk production. Many of these hormones from the anterior pituitary have as their target other glands, and so we call these effects tropic effects. So they're going to direct the synthesis of another hormone. So this is increasingly complicated hormonal pathways. So for example, FSH, follicle stimulating hormone, and LH, luteinizing hormone, are going to have their effect on the testes or ovaries and direct them to release other hormones from these glands. Similarly, with thyroid stimulating hormone, it's going to have an effect on the thyroid gland, and ACTH is going to have an effect on the adrenal gland. Some of them have non-tropic effects, so they're going to affect their target cells directly rather than by releasing another hormone from them. So examples are prolactin and melanocyte stimulating hormone are going to have their effects directly. And then growth hormone has both tropic and non-tropic effects, so it can affect tissues directly, but it can also have glands as its target. So let's take a look at an example of one of these more complicated hormonal pathways. Sets of hormones from the hypothalamus, anterior pituitary, and a target endocrine gland are organized into a hormone cascade, or a pathway, a more complicated pathway of hormonal signaling. So in humans and other mammals, thyroid hormone regulates many functions in the body. And if the level of thyroid hormone drops in the blood, this is detected by chemoreceptors within the body, and the hypothalamus will respond by secreting thyrotropin-releasing hormone. So this is a tropic hormone that's going to affect another gland, which is going to cause the anterior pituitary to secrete thyroid-stimulating hormone. So here we get the signal of low thyroid hormone. The neurosecretory cells in the hypothalamus are going to secrete TRH into the blood. The blood's going to go directly to the anterior pituitary, which is going to secrete thyroid-stimulating hormone. This is going to circulate through the body until it reaches its target in the thyroid gland. So it's going to stimulate the thyroid gland to produce thyroid hormone, which is then going to circulate through the body, find all of its targets, and complete its various functions. But the thyroid hormone is going to increase in the body tissues, sending a signal back through this negative feedback loop to the hypothalamus and also directly to the anterior pituitary that we now have enough thyroid hormone, and it's going to shut down the production of thyroid stimulating hormone and thyrotropin releasing hormone. So if this thyroid hormone production is disrupted, this can result in very serious disorders. So thyroid hormone is one of the only substances in the body that relies on the element iodine. Iodine is part of the molecules that compose thyroid hormone. So each of the different kinds of thyroid hormones contain at least a couple of atoms of iodine within them. So iodine can't be obviously synthesized in the body. It has to be taken in through dietary sources. So in general, the ocean is very rich in iodine. So if you eat a lot of seafood, will supply all the iodine you need. In many places, the table salt that we use is actually supplemented with iodine. In case you're in a part of the country that doesn't have ready access to seafood, the iodine in the salt provides all of the iodine that's needed to produce these thyroid hormones. So if you don't get enough iodine in your diet, there are going to be low levels of thyroid hormone. It's not going to be able to be synthesized without that iodine. And the pituitary is going to continue to send the signal to the thyroid gland to produce more thyroid hormone. And so this causes the thyroid to enlarge, resulting in a condition called a goiter. So remember that the thyroids are located in the neck, and so this goiter is going to present as kind of a swelling around the neck, and it can be very minor in the early stages. So here we just see a small swelling around the neck, but if this continues untreated for long periods of time, this can have very, very profound effects on a person's appearance. Here's an example of a very large goiter in this man. Hormones also regulate growth. So there's a particular hormone called growth hormone, which is secreted by the anterior pituitary gland. And as we mentioned before, it has both tropic and non-tropic effects, meaning that it's going to have as its targets both other glands that it's going to direct to secrete other hormones, and it's also going to have direct effects on other target organs. So the liver is a major target and responds to growth hormone by releasing insulin-like growth factors, and these insulin-like growth factors are going to stimulate bone and cartilage growth to allow proper development during childhood. So an excess of growth hormone can cause a condition called gigantism, so growing out of control. This can happen either during early development, in which it causes excessive growth, but still generally normal body proportions, or it can also happen later in life, in which it causes increased bone growth, particularly around the face and hands and feet. And conversely, if there is a deficiency in growth hormone, this can cause dwarfism. If this is detected early enough during the growth period, this can be treated with synthetic growth hormone to attain sort of normal heights, but otherwise this can result in dwarfism, in which case this form of dwarfism, the body is normally proportioned but just undergrown. So in this web lecture, you've reviewed cell signaling in general, these different transduction pathways. You've been introduced to the different kinds of pathways hormones can take through invertebrates and vertebrates. Now we're going to, in the next web lecture, look in more detail about how these different pathways work to regulate different systems in the body. So we're going to see how these pathways will have a regulatory effect on homeostasis, maintaining conditions in the body within optimal levels, and also their effects on development and on behavior.